We're ready to pick up with part 5 of 6 parts on intracellular membranes, cellular organelles, and vesicular traffic. And what we're going to do in this part of the presentation is focus on vesicular transport in relation to the membrane flow family of organelles. So we're going to be looking at movement of material by way of vesicles from the ER to the Golgi and through the Golgi, and then to secretory vesicles and to the cell surface. We'll also look at routing to lysosomes a little bit, and we'll talk a little bit about the endosome compartment. Before we do this, let me just go over this slide. It's kind of a reminder of what we talked about very early in the first presentation in this set. We're considering vesicular transport. What we're considering is something moving from a donor compartment to a target compartment. We're considering a membrane vesicle that's pinching off, and as it's pinching off, it's trapping some type of cargo to move, and this vesicle often will pinch off as a coated vesicle. It's going to travel in the cytoplasm, usually on cytoskeletal components. It will lose the membrane coating before it fuses with the target compartment. When it fuses, of course, the cargo is delivered to the target compartment. Now, the diagram is colored in two different ways so that you can see the outer and inner bilayers of these organelle compartments so that you appreciate that the membrane topology is maintained. Let's consider the Golgi for a minute. Here's an electron micrograph of the Golgi, and this is actually an electron micrograph from a plant cell, simply because plant cells have really elaborate and substantial Golgi, and it's easy to see them. But what we say for the Golgi, it holds for any cell type you're looking at. When we consider the Golgi, we consider the Golgi a series of stacked membrane compartments. So unlike the ER, which we think of as one continuous compartment in the cytoplasm, the Golgi, we believe that each stack acts as a different compartment. So we can broadly talk about a cis face of the Golgi and a trans face of the Golgi. The cis face you can think of as an entry face or a forming face from the Golgi. That's where the Golgi is receiving material by way of vesicles or membrane tubes from the ER. And the trans face of the Golgi you can think of as the exit face or maturing face of the Golgi. That's the face where vesicles are budding off of the Golgi to go someplace else, either, say, to a lysosome or to become a secretory vesicle. We can define regions of the Golgi per se. So people will talk about a cis Golgi network and a trans Golgi network, and people will also talk about the Golgi cisternae proper. So one might talk about cis cisternae, medial cisternae, and trans cisternae. And I think it makes sense if you just think about it in orientation of the Golgi. Cis face is the entry face of the Golgi, that part of the Golgi that receives material from the ER. The trans face of the Golgi, the exit face, is that face of the Golgi where vesicles are budding off to go someplace else. If we look at protein pathways in the ER and in the Golgi, this is a fairly complicated slide. First of all, what we can say by review, there's co-translational transport of proteins into and across the ER membrane, and this is what we've already talked about. Vesicles will then bud off of the ER, and they will form what's called a cis Golgi reticulum. And then there'll be anterograde movement through the Golgi, and by anterograde movement we mean a movement in a forward direction. And by a forward direction I mean if you are envisioning exocytosis or the biosynthetic pathway for exocytosis, the anterograde movement would be relating to material that's packaged to go to be fusing with the cell surface or to be excreted from the cell. You could also think of anterograde movement as something going through the Golgi to be routed to lysosomes. So that's anterograde movement. In many cases, we think that depends on movement by what's called cisternal progression. Anterograde movement is dependent on COP2 coated vesicles and tubes. If we look at regulated secretion, or we look at lysosome formation, so regulated secretion or lysosome formation, the vesicles that are involved here are involved initially with clathrin. If we are looking at constitutive secretion, where something coming from the Golgi fusing with the cell surface for excretion, but not requiring any signal, those vesicles are coated with COP2 proteins. So let me reiterate that. Regulated secretion is under the direction or response to some signal, and the vesicles form with clathrin. Constitutive secretion, no particular signal is involved. The vesicles are always forming. There's always a basal or low level of secretion, and those vesicles are forming with COP2 proteins. Either regulated or constitutive secretion, that's talking about anterograde movement. If we talk about retrograde movement, we're talking about movement in the opposite direction. So if you were to follow vesicles from one face of the Golgi going in the reverse direction, that retrograde movement depends on COP1 coated vesicles. So you want to keep straight COP1 vesicles, COP2 vesicles, and clathrin coated vesicles. I'm almost certain you'll see questions like that on shelf exams and board exams. So it's something that people that write those exams, they like to be picky about those details. If one looks at the Golgi dynamics, we have COP2 vesicles budding off the ER, and they are fusing to form this ER Golgi Intermediate Compartment or cis Golgi network, that we can consider that the same thing, the ER Golgi Intermediate Compartment or cis Golgi network, vesicles fusing from the ER and coalescing to form this structure, or alternatively, these vesicles are moving directly to the cis Golgi cisternae. One of two ways that we think material might get to the Golgi. Movement of these vesicles depends on microtubules and microtubular motor proteins, and we'll talk about microtubule motors in a little more detail later in the course, but you just have to appreciate that movement from the ER to the Golgi, and indeed to maintain a functional Golgi, depends on an intact microtubule cytoskeleton. Here, showing retrograde movement going in a reverse direction, COP1 coated vesicles forming, and here the retrograde movement with COP1 coming back through the various stacks of the Golgi, like so, or coming back from the Golgi all the way back to the ER, perhaps passing through the ER Golgi intermediate compartment on the way. That retrograde movement depends on COP1 vesicles. Here we're talking about the endosomal and lysosomal pathways, and these usually depend on clathrin. Here we're looking at a clathrin-coated pit forming at the cell surface, pinching off to form a clathrin-coated vesicle, not unlike what we talked about with LDL internalization. When this vesicle has pinched off, before it can fuse with an endosome, it has to lose the clathrin coat, and then it fuses with an endosome. Some portion of this endosome might break off and go to a lysosome. Other portions might break off and return to the cell surface. So you would imagine, again, if it was the LDL receptor, a component of the endosome might break off and bring back to the cell surface the LDL receptor itself, whereas another component might break off and bring the LDL particle to the lysosome so that it could be broken down into free cholesterol. we have said that movement of vesicles depends on an intact cytoskeleton, something that is relatively new that cell biologists are beginning to appreciate. Movement of the vesicles, and especially as the vesicles get towards the donor compartment, might actually depend on the interactions with actin. So the thought might be that all the way through the cell there'll be microtubule networks that vesicles will travel on, but the microtubule networks we know don't actually touch the cell membrane. We do know that actin seems to get an intimately close association with the cell membrane, and so what people are beginning to think is that vesicles might travel along microtubules and then will come off the microtubule roadway, if you want, and hitch a ride or jump onto actin filaments to make the final bit of migration to the cell surface. And it's possible that the interactions of the vesicles with actin might play some role in the fusion of the vesicle with the target compartment. This is an example of movement of vesicles from the ER to the Golgi along microtubules, and we'll talk about this in just a second, but first I would like to highlight this concept of a vesicular tubular cluster, and I'm kind of tracing that right now with my pointer, this vesicular tubular cluster. It has been suggested that conglomeration of membrane tubes has a higher capacity to move cargo than just simple membrane vesicles. So the thought being that this vesicular tubular cluster may be a more efficient way to move a large amount of cargo. The membranes may be enriched with membrane proteins in relation to soluble cargo, so possibly a more efficient way for moving material. But now, what this part of the image is showing is this vesicular tubular cluster associated with a microtubular motor protein. Now, we haven't yet talked about it in the course, but we will later in the course. Microtubules have a polarity. They have a plus end and a minus end. And the plus end of these microtubules orients themselves always from the ER going towards the Golgi. And these microtubular motors that move material on the microtubules are called kinesins. They move material towards the plus end of the microtubule. In the same token, if you were to think of retrieval of vesicles from the Golgi back through the Golgi stacks, maybe all the way back to the ER, this retrieval of vesicles, the vesicles are pinching off with COP1 proteins, but they would also move on microtubule tracks. It's just not drawn here on the bottom of the slide, but the microtubular motor then would be dynein. Dynein is a microtubular motor that moves cargo towards the minus end of the microtubule, kinesin being a microtubule motor that moves things to the plus end of the microtubule. One more point made from the slide. This is anterograde movement that I'm tracing like so. Anterograde movement with COP2 coated vesicles as the vesicles initially form, and retrograde movement this way, back towards the ER, through the Golgi back towards the ER, with COP1 coated vesicles forming to start with. Remember this vesicular tubular cluster, because here is a real electron micrograph of this vesicular tubular cluster, and I am trying to trace it a little bit. And as you see, this looks suspiciously like the cartoon. So these vesicular tubular clusters may actually be real structures. And so we now appreciate that transport through the Golgi can be described by two different models. These models are not mutually exclusive, and indeed movement through the Golgi, movement from the ER to the Golgi and through the Golgi, may be a little of both models occurring. So in the top we're looking at vesicular tubular transport, so vesicles budding off the ER, coalescing into something like the cis-Golgi network, and this vesicular tubular cluster then moving forward in an anterograde movement to the Golgi. And then the thought being that vesicles will bud off of each stack of the Golgi in an anterograde movement. So this vesicle budding off would be with COP2 proteins. And then retrograde movement recycling back through the Golgi, vesicles budding off from one face and going to another face or another cisternae, and this would be with COP1 coated vesicles. So that's vesicular tubular transport model, and that can contrast a little bit with the cisternal maturation model. Here the model would say that vesicles form from the ER, coalesce into the cis-Golgi network, forming this vesicular tubular cluster, and then the cis-Golgi network moves forward, and then in an anterograde movement, rather than individual little vesicles pinching off, an entire Golgi cisternal stack, the entire stack moves in an anterograde movement in a forward direction. Again, all of this would be on microtubule tracks with microtubule motors, and then the recycling would be by vesicles pinching off and going in a retrograde movement. So the key with this one is each stack of the Golgi moves forward and a new stack comes in and takes its place. So the models are not mutually exclusive, and it may be that in real life different components of each model will explain different metabolic conditions in a cell. Here is some views of the Golgi. Here's an unstained view of the Golgi. Here's a view of the Golgi that's showing the cis region of the Golgi stained. Here's a view of the Golgi showing the medial region of the Golgi stained, and a view of the Golgi showing the trans-Golgi network stained. You don't need to really worry about what these stains are or how the staining is done, but you could imagine if you're looking at movement through the Golgi, you're moving this way into the cis-Golgi, the medial Golgi, and the trans-Golgi. So here is a cartoon that will look at functional compartmentalization of the Golgi, and most cell biologists that have studied the Golgi look at the Golgi as having three major divisions where there are distinct biochemical functions that occur. So let's start at the top of the slide. This would be the ER, and so vesicles moving from the ER to the Golgi. In the cis-Golgi network, it's thought that there'd be phosphorylation of oligosaccharides on proteins that are destined to be routed to lysosomes. In the cis-cisternae of the Golgi, mannose residues are removed. Remember, we said that glycosylation, you get these sugars that are added all at one time, and then these sugars are clipped and processed as you move through the Golgi. So in the cis-Golgi, it's thought that one removes mannose. In the medial cisternae of the Golgi, there's removal of mannose and addition of N-acetylglucosamine. In the trans-cisternae of the Golgi, there's addition of galactose. In the trans-Golgi network, there's addition of sialic acid. So it's thought each of these cisternae of the Golgi has unique biochemical functions. So the question becomes, how can this unique biochemistry in each function be maintained, especially if you look at the cisternal progression model of movement through the Golgi, where an entire stack moves forward? Because the thought would be that if this stack is moving forward, you're eventually, for example, losing the functional ability to remove mannose or the addition of being able to add N-acetylglucosamine. But if the stack is moving forward, how come the enzymes are not still being active? So that's kind of an interesting thing that people have tried to consider. So put in another way, cisternal progression, the question becomes, how is the biochemical uniqueness of each Golgi stack explained? in relation to vesicular transport, one could also ask, how do ER resident proteins get returned to the ER so the ER remains functional? Because you can imagine that if you've got something in the lumen of the ER that's going to bud off and carry a cargo to the Golgi, it may be budding off and taking with it some of those proteins that were necessary for co-translational translocation of proteins in the ER membrane and the rough ER membrane to start with. So the questions then, how is the biochemical uniqueness of each Golgi stack explained with cisternal progression? And if you're talking about vesicular transport, how does the ER remain functioning if you're potentially taking resident proteins away from the ER as they're moving forward with vesicles? So let me just remind you of this little cartoon that we showed very early in our first presentation. Donor compartment, target compartment. So you're capturing the target, the little red vesicles, and maybe these little black lollipops are just moving by just randomness, just stochastic events as the membrane is pinching off. But what if these little black lollipops are necessary for the function of the ER? How do you maintain the function of the ER if you're taking away some of these proteins? So those are the questions that one is asking. And so what we've come to understand, there's a salvage pathway for the ER or for Golgi proteins that relates to the compartment called the ER Golgi Intermediate Compartment. And we know now this involves K-del sequences and K-del receptors. We know that it involves COP1 proteins. We know it involves movement of vesicles on microtubules, and we're pretty sure that there's likely to be a little bit of a pH gradient in the vesicles. So here is the scenario that's established. You have a soluble ER resident protein. This is a protein that should be in the ER, but it's in a Golgi stack because of anterograde transport of vesicles or tubes. Well, what is thought is that these ER resident proteins have this K-del sequence that we mentioned in one of our earlier presentations in this series, as this K-del sequence, and there are also K-del receptors that are in various parts of the Golgi. The K-del receptors would have receptors that would specifically recognize lysine, aspartic acid, glutamic acid, leucine, this so-called ER retention sequence. And so what this scenario is showing is this protein with the sequence is like the ligand, this being the receptor. So the receptor is binding the ligand, and now a vesicle is pinching off, coated with COP1 proteins, because this is for recycling. And so here are these vesicles that are pinching off and moving back down through the faces of the Golgi and maybe back to the ER. So here's the scenario in a little more complex picture. The little yellow diamonds are secretory proteins that should normally be part of, let's say, extracellular matrix material. Maybe it's like a collagen protein. Here are these ER resonant proteins that are in the ER. Here, by constitutive secretion, because most extracellular matrix components are secreted constitutively. COP2 vesicles pinch off. Here's one pinching off with just the cargo that needs to be transported, so it's going through the Golgi. Never how it moves through the Golgi is going to get to the trans-Golgi network to bud off. But here is a vesicle pinching off in the Golgi. It's just in the ER, rather. It's just random. It's taking the secretory cargo, but also by chance picking up some of the ER resonant proteins. Well, lo and behold, if those ER resonant proteins have this K-del retention sequence on it, and then here are the K-del receptor proteins, well, they're all set. They can come in and bind the ER resonant proteins and can allow recycling. So recycling all the way potentially from the trans-Golgi network all the way back to the ER, and this is on microtubules. What we do know is if you look at the pH in the luminal compartment of the ER and of these vesicles and of the Golgi cisternae, as you move more towards an anterograde direction, you get a relatively lower pH. Not significant, but a relatively lower pH. So there's some thought that maybe the pH is involved in how the binding of these K-del receptors with the retention sequence, that may have some involvement in enhancing that binding. The return of these vesicles through the Golgi or all the way back to the ER will depend on microtubules with cytoplasmic dynein motors, because you're going to be moving in a retrograde direction towards the minus end of the microtubules. What if recycling, instead of going all the way from transport of the Golgi all the way back to the ER, what if recycling only went part way? In other words, you only went through part of the Golgi and then the recycling stopped. That recycling partway through the Golgi may be the way to explain the biochemical uniqueness of each Golgi stack. So the enzymes that are involved in certain regions of the Golgi, maybe they recycle back to those regions all the time, so that even though an entire cisternae is moving forward, the appropriate enzymes that are needed for function in the previous Golgi cisternae are brought back. That's an interesting model and an interesting extension of what we think is occurring. Here is just last little statement I want to make. You can actually dissect movement through the Golgi, and you can look at an intact Golgi and see what happens. Here is a drug called nicotazole. Nicotazole is a drug that interferes with microtubule assembly. If you treat cells in culture with nicotazole, you disrupt microtubules, and so you block the recycling pathway. The ER eventually loses function, and the Golgi eventually loses function. If you block anterograde movement with a drug like Brifeldin A prevents the assembly of COP2-coated vesicles. Brifeldin A will inhibit constitutive secretion. It will also inhibit early movement through the Golgi stacks. So these two drugs can, in appropriate doses, disrupt the Golgi. Now, if you treat with high levels of nicotazole or with other microtubule poisons that totally disrupt microtubules, you'll totally disrupt the function of the Golgi. So we know for sure that an intact Golgi requires an intact microtubule cytoskeleton.